Progress.
Hello friends, myself Dr. Amit, and today I'm going to teach you about the sliding filament theory of the muscle contraction. I already taught you about the sarcomere, and if you're not through with the sarcomere topic, I request you that you first learn the sarcomere structure. If you don't know what is A filament, what is actin filament, what is myosin filament, what do you mean by M line, what do you mean by Z line, then you won't able to learn this topic. So first, what is the requirement that you need to learn about the sarcomere, and only then you go with this video lecture. So let me present the slide. The just a second. Okay. Now we can see. Okay. So uh, what I have kept before you is the muscle contraction slide. So as I said earlier, that you must be through with the structure of sarcomere that I'm not going to repeat here. Right now, what I am showing you is the contraction mechanism, sliding filament theory. The brown color structure, what here is represent myosin filament. This is the thick vein. And this is the individual myosin molecule, tail part and the head. Then this structure, yellow dotted structure is the actin molecule. That is the actin filament. Many globular structure unite to form actin molecule, and many actin molecule make the I beam. That is the thin filament. Many actin molecules unite to form the thin filament, and this structure, thread-like structure that is a little darker yellow, is the uh, tropomyosin complex. What does it do? Tropomyosin complex hide the myosin binding site of the actin molecule. And how this happens? To remain in the contact, these tropomyosin molecules held in their position by another molecules, and that is called the troponin molecules. So these troponin molecule, that is this gray color is the troponin molecule. So when this troponin molecule remain attached with the tropomyosin, it is not able to. In that case, tropomyosin remain in its position, and that causes the myosin binding site on the actin molecule remain closed, or they remain hide. So myosin molecule cannot attach them. So like troponin is like a nail. It keeps the tropomyosin at its place. So if you remove the troponin, what will happen? Tropomyosin get disoriented and the myosin's binding site get exposed. So this is the fundamentals. Now let's talk about this myosin molecule. Now these myosin molecules have two features. As you know, this is the tail part and this is the head part. The head part has got two regions. One domain is the binding site for the with the troponin. Another, of course, there's a domain for the binding site for the actin. Then it has the binding site for the ATP also. One, it has binding site for the actin molecule. Then second, it has got binding site for the ATP molecule. And the third, it has got the binding site. It has got the ATPase activity. So myosin head is a sort of enzyme. It is ATPase and it has got a one domain, one protein molecule structure that acts certain sequence of the domain means certain amino acid region that act as the ATPase enzyme. So what happened? How they start? So first of all, what happens? Calcium molecules. When the calcium influx is there, when the muscles is actually given signal from the nerves to get contract, what happened? That calcium ion get released from the endoplasmic reticulum, that is the sarcoplasmic reticulum. Now what happens? This calcium ion has got binding site with the troponin. This calcium ion has got the binding site with the troponin molecules. So what happens? Troponin has binding site for the, it has got it's actually nailed the troponin tropomyosin to its place. That is this gray color structure. But this gray color structure, that is the troponin, has also has the affinity for the calcium. So when the contraction is there, sarcoplasmic reticulum releases the calcium ion. Now this calcium ion get attached with the troponin complex. So calcium will attach here. So what happens? Now attached with the calcium ion, it get detached. It get detached, or it get you can say disorient from this its place. So now what happened? As the troponin get disoriented from its place, tropomyosin also get disoriented, and that lead to actin molecules, myosin binding site get exposed. What happens? This dotted structure is the myosin binding site. The brown dotted structure on this yellow molecule is the myosin binding site. Again, I am repeating what happens. Calcium ion gets entered. This calcium ion comes out from the endoplasmic sarcoplasmic reticulum. It binds with the troponin molecules. Troponin molecules get displaced. As it get displaced, in turn, troponin get displaced. In turn, tropomyosin get oriented from its place, and that leads to exposure of the myosin binding site on the actin molecules. Now, what will happen? Now, what will happen? Now, with now what will happen? That uh, now the role of ATP. Now this comes the ATP. If the calcium is not there, this will what happen? Now what happen? ATP get attached to its ATP binding domain, and then ATP gets converted into ADP and phosphate. Now ADP and phosphate, and these in this form, these get excited. Again, I'm repeating, myosin has got ATP binding site, it has got the ATPase activity, and it also got actin binding site. Now, when the calcium influx is there, what happened at the myosin level? ATP get attached to the myosin molecule, and it get hydrolyzed into ADP and P. This is called the myosin hates at hydrolyzed ATP and become the reoriented energized. So at this stage, when it get into ATP gets converted to ADP and P, we say that myosin head is energized. After getting energized, it is now ready to contract the muscle. It is ready to attach with the actin molecule and get it passed to the stroke it toward the center, that toward the M line. So now what will happen? This energized myosin head will first release the P molecules and get attached. ATP hydrolyzed produce ATP and P, and when this hydrolyzed phosphate molecule detached, and in second step, what will happen? This myosin molecule along with ADP attached to with not ATP, not the phosphate, but ADP remain attached, and then is this stage, this attached with the actin molecule. Now, clear? Phosphate is released, but now what will happen? ADP still remain there, and the myosin molecule attached with the actin molecule. This is the call. At this stage, we use the term cross breach. Till now we say myosin head. Now when they attach with that, that is called the cross bridge. So what will be structure? What will be scenario at the cross bridge? Phosphate is not there, but ADP is there. And then these get attached with that. And then comes the power stroke. And then at first of all, it get energized, then get attached, and then pulling will take place. So pulling of the actin molecule to take place, what will happen in the third step? This ADP molecule get detached from the myosin head. At the third step, these ADP molecule get detached from the myosin head, and that will carry out the pulling of the sliding of the actin molecules. So these way, many such molecules of myosin molecule will carry out the pulling of the sliding filament toward the center, that is toward the M line. So what happens from both sides? Myosin will take these. Suppose you have to carry out this particular exercise in the gym and you're pulling the rope toward the center. So that way, both sides actin molecules get crossed, they pass to the center, and sometimes so Z line, you can say Z line, the Z and Z disc of the both sides come closer, and that length of the Z line gets shorter. The length of the Z guide gets shorter. Keep in mind that individual actin molecules or myosin molecules do not get change in the size. They only pass, they only overlap. So that called the contraction shortening of the Z disc. The shortening of the Z disc, and this way entire muscle fiber shortening of the Z disc take place. Now, as the shortening of the muscle fiber take place, the same mechanism follow to entire muscle, so shortening of the muscle, and that is the contraction. Now, I will make it more clear with the reading material with you. So I will now, after briefing this, I will go with the slide wise. Again, I am showing this diagram. First of all, let's again go through this diagram. First, myosin head hydrolyze ATP and become reoriented, energized. So this is the energized molecules you can see. And then it has got ATP converted to ADP and P. ATP and P. ATP when gets hydrolyzed to ADP and P, that makes the myosin molecule energized. In step 2, myosin get attached to the actin filament, and at that place to happen, P is released. At this stage, ADP is still there. When ADP is released, then we have the next stage. When ADP is released, then there is a power stroke. At the power stroke, it will pull toward it. Now, after the pulling, when its task is over, it still remains attached. After contracting, it doesn't detach. After contracting, it doesn't detach, but it remains firmly held along with the what you call actin molecule. So to get detached from the actin molecule, what is the next stage required? It cannot carry out the next again and again. So again, for pulling out again the actin molecule, what is required? Again, one more ATP molecule. So until the new ATP is available, it remains attached. Now what happened? When it remains attached, at that time itself, another ATP molecules is get attached to the ATP binding site. Am I clear? Let me repeat. Up till now, you are clear that ADP release and then carry the power stroke. Now, what happened? Power stroke has happened. Suppose this is the initial play, and at least their power stroke has happened. Now, it does not get detached from this actin molecule, but it remains attached. So, how get it detached? For what happened? What is required to get detached from the actin molecule? So, it is required ATP molecule. So, now what happened? When new ATP molecule, say for example, bind at this side, say for bind this side, then it get detached. So when ATP molecules bind this side, it get detached itself, the head get detached from the actin molecule, and again cycle is repeated. Then again ATP is hydrolyzed into ADP and P, and again it carry out the next stroke. So this way multiple stroke causes the muscle contraction. So let me go with the slide wise so it will be more easier for you. Okay, so muscle contraction occur because myosin heads attached to the end walk along the thin filament at the both end of the sarcomere, progressively pulling the thin filaments toward the M line. As I already spoke about this, they keep the thin filament toward the center M line. As a result, the thin filament slides inward and meet the center of a sarcomere. They may even move so far inward that their ends overlap. Both sides thin filaments and get overlapped. As the thin filament slides inward, the Z disc comes closer together and sarcomere size gets shorter. However, the length of the individual thick and thin filament do not get changed. Shortening of the sarcomere causes the shortening of the whole muscle fiber, which in turn leads to the shortening of the entire muscle. So I have already spoken about this. Now this is the four-step contraction cycle. What happened at the onset of contraction? How does it begin? So signal is the calcium ion release. The sarcoplasmic reticulum releases calcium ion into the cytosol. There they bind to troponin. Step 1, sarcoplasmic reticulum releases calcium ion. This calcium ion gets attached with the troponin complex. Troponin then moves tropomyosin away from the myosin binding site on actin. Once the binding sites are free, the contractile cycles begins. That is the repeating sequence event that will cause the filaments to slide. The contraction cycle has 4 steps. That's us here. I have already gone through this diagram. Now let's go with the writer. So first step is the ATP hydrolysis. The myosin head include an ATP binding site and an ATPase enzyme that hydrolyzes ATP into ADP and a phosphate group. This hydrolysis reaction reorients and energizes the myosin head. So again I said ATP attached with the myosin. ATPase enzyme carry out the conversion two-step reaction. ATP binds to the myosin head. As it binds, ATP gets converted to ADP by P by the enzyme called ATPase. Now, both the ADP and phosphate group remain attached. The myosin head remain attached with the myosin head. At this stage, we say the myosin head is energized. Now, the next step is to energize the myosin head will get attached to this energized the ADP state. It get attached to the actin molecule. But before that, what will happen to get attached? Phosphate will release. So now in the second step, attachment of the myosin to actin to form the cross bridge. In this step, the energized myosin head attached to the myosin binding site on the actin and release this the previously hydrolyzed phosphate group. When the myosin head attached to the actin during contraction, they are referred to as the cross bridge. So I have already spoken about this. Now the third step is the power stroke. After cross bridge form, the power stroke occurs. During power stroke, the site on the cross bridge where ADP is still bound open. So now ADP gets dislodged. As a result, the cross bridge rotates and releases the ADP. The cross bridge generates a force as it rotates toward the center of the sarcomere, sliding the thin filament past the thick filament toward the M line. Now, detachment of the myosin from the actin. At the end of the power stroke, the cross bridge remains firmly attached to the actin until it binds another molecule of ATP. As the ATP binds to ATP binding site on the myosin head, the myosin head detaches from the actin. So this way, the entire contact you can see. This is the relaxed muscle and this is the contact muscle. You see the Z line area. You can see this power stroke causes a Z line. This is you can see how much overlap is there. You can see here actin molecule very far away, but you can see very much toward the Z line, and sometimes even they can overlap with each other. So this is the M line and this is the Z line. Now, what in this diagram is shown, this is a relaxation state. When there is no signal from the neurons, there is a relaxed state, there is no more calcium available. All the calcium through this channel, calcium active transport from will from all cytosol will again remove from the cytosol and get back into this sarcoplasmic reticulum, that is a tank that stores the calcium ion. So through this channel, calcium will move in, and for that reaction, there is a calcium active transport. So this is the calcium active transport that get back the calcium pool inside the sarcoplasmic reticulum. There is no calcium, then troponin remain firmly bound to tropomyosin, and tropomyosin holds to its position, and then there is a hiding of the myosin binding site. As the myosin binding sites are not exposed, the myosin molecule cannot bind to the myosin molecule cannot bind to the actin molecule, and muscle remain in the relaxed status. And during the contraction, what will happen? I have spoken that the side from the endoplasmic reticulum, that is sarcoplasmic reticulum, calcium through this channel is open, calcium released, and then it bind with the troponin molecule, and then I have already spoken that contraction comes into the action. Now gets further. So the contraction cycle repeats as the myosin ATP is hydrolyzed. The newly bound molecule of ATP and continue as long as ATP is available and as long as calcium is near there in the vicinity of the thin filament. So as long as calcium is available, as long as ATP is available, there will be contraction. The crossbreak keep rotating back and forth. The crossbreak, that the myosin molecule keep back and forth rotating with each power stroke, pulling the thin filament toward the M line. So with each stroke, M line is thin filament, pulling the thin filament toward the M line. And there is not only one molecule. Each of the 600 crossbreaks in one thick filament. I'm talking about only one thick filament. There's so many thick filaments. Each of the 600 cross bridges in one thick filament attach and detach about 5 times per second. You can imagine 600 cross bridges in one thick filament attach and detach about 5 times per second. At any one instant, some of the myosin heads are attached to actin, forming the cross bridge and generating the force. Another myosin heads are detached from the actin, getting ready to bind again. So at any particular point, there are certain molecules get attached and certain remain detached. So constantly they are pulling the actin molecules. As the contraction cycle continues, movement of cross bridge apply the force that draw the Z disc toward each other and the sarcomere shortens. During a maximal muscle contraction, the distance between two Z lines can decrease to half of the resting length. So you can get shorter in length. Z disc comes in so closer, the sarcomere size gets shorter, half to its initial size. The Z disc in turn pull one on neighboring sarcomere and the whole muscle fiber shorten. Some of the component of the muscles are elastic. So now you can study from the slide. They stretch slightly before they transfer the tensor generated by the sliding filament. As I taught you in earlier my lecture, that elastic component to the titin molecules. In my previous lecture, I had taught about this titin molecules. Connective tissue and the muscle fibers, that is endomysium, perimysium, all have elastic component. And your tendons that attach the muscle to the bone. As the cell of skeletal muscle start to shorten, they first pull up there on the contractive tissue covering the covering and tendons. So entire thing get transferred to level of the epimysium, endomysium to a tendon. The covering of the tendon stretch and then become taut. Taut matlab exactly very tightly strengthened tightly. It becomes very taut and the tension passes to the tendon, pulls toward the bone, and that tendon pulls the tension pass to the tendon, pulls on the bones to which they are attached. So entire muscle pull the bone to which they are attached, and the result is the movement of the body part. So all this we have already talked about. So extraction and contraction, you can just go through this slide. Increase concentration of the calcium, and then how it comes out from the sarcoplasmic reticulum. What does it cause? You can go through the slide by your own. We already talk about this. Okay, clear? Now, as the calcium ion contraction of the cell rise a tenfold or more, the released calcium combined with troponin, causing it to change its shape. This confirms the change moves topomyosin away from the myosin binding site on the actin. Once these binding sites are free, myosin has bind to them and form the cross bridge, and the contraction begins. So again, I am repeating the same thing. The event just described constitute the excitation, countercoupling that steps the connect the excitation, the contraction. Again, you can go with this slide. Okay. You can take a snap of this. So when in the relaxed status, calcium ion concentration 10,000 times higher in sarcoplasmic reticulum than the cytosol. And then at that stage, there will be relaxation will occur. So hope you have understood this topic. It's very easy topic, but what is the pre-requirement that you need to have knowledge of the sarcomere. This is the diagram, entire cycle diagram. Excitation, step 1 excitation, cross bridge formation, then pulling, and then detachment. So in these 4 ways the cycle gets continuous. Okay, I hope you have understand. And here, so hope you understood the topic. So here, if you have any doubt, you can contact me to email. Here, sliding filament theory gets over. You can add for some description about this discovery. There are two group of scientists. One had done their work on the frog, and another group of scientists, two group of scientists had done their work during the same period of in the same sort of time. Another group of scientists had done their work on the rat muscles. So these you can find from the reference books. The name, I'm not mentioning their names. So they have given the sliding filament theory. Hope you understood the topic and good luck for your exam.